These pigments are organized into two discrete photochemical light harvesting complexes. Okay, what are they? Photosystem 1 and photosystem 2. Second one is photosystem 2. In the PS1 and PS2, in the photosystem 1, okay, 700 nanometers of light is required for the reaction. In the PS2, 680 nanometers of light is required for the reaction center. So, light sequence based on that, they are classified into two types. Light harvesting complex, which is there around the photosystem 1, made up of more than 100 pigments. More than 100 pigments are there around the light harvesting complex within the photosystem. Now, how it will happen, let us see. If you see the photosystem 1 here, these are called the light harvesting centers or light harvesting system that contain more than 100 pigments. Let us imagine sunlight. Okay, when the sunlight falls upon the light harvesting system and photosystem 1, okay, 700 nanometers of light is utilized by the PS1 or photosystem 1. So, when the sunlight falls on it, as you know that sunlight contains photons, energy packets called photons. When these photons are reached to the light harvesting complexes, immediately 100 pigments along with photosystem 1 accelerated and the PS1 accelerated releases 2 electrons outside. These 2 electrons are moving forward, okay, reaching to primary electron acceptor, that is ferredoxin. What is that? Ferredoxin. So, when the electrons, primary electron acceptor in the cyclic photophosphorylation, if you take the light reaction is classified into two types. First one is cyclic photophosphorylation and the second one is non-cyclic photophosphorylation. Now we are learning the first one, cyclic photophosphorylation. In the cyclic photophosphorylation, primary electron acceptor is ferredoxin. Through ferredoxin, electrons will be reached to the cytochrome complex or cytochrome B6. While the electrons are moving from cytochrome B6 to cytochrome F. Okay. Here what happens, amount of energy will be captured by the ADP and which helps to form the ATP. Okay. During this process, electrons are moving in the downward direction or downward hills. In this case, while they are moving to downward hill, they releases the energy. The released energy is in the form of phosphate and will be converted into ATP. From the cytochrome B6, electrons moving into cytochrome F, here phosphate is captured and phosphate is captured by the ATP, which helps to form the ATP. From the cytochrome F, finally 2 electrons will be reached to the light harvesting complex and photosystem 1. Like this, cyclic photophosphorylation helps in the synthesis of ATP while the electrons are moving in the downward. Finally, you can see light harvesting complex made up of more than 100 pigmented molecules. Here, reaction center will be there. Okay, primary electron acceptor will be ferredoxin. After this, each photosystem has all the pigments except one molecule of chlorophyll A. Okay, these pigments helps to make photosynthesis more efficiently by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. Okay, we have seen there photosystem 1 and 2 formed by the chlorophyll A. The reaction center is different in both the photosystems. As I told you, photosystem 1 activated when there is a 700 nanometers of light, while PS2 it has absorption maximum at 680 nanometers of light, hence it is called P680. Finally, electron transport. In photosystem 2, a reaction center, okay, a reaction center of chlorophyll A absorbs 680 nanometers of light or wavelength or red light, causing electrons to become excited and jump into outer orbit from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them an electron transport system that contains cytochrome complex. Now, this movement of electrons in downhill in terms of helps in oxidation and reduction or redox potential scale. Now, oxidation reactions and reduction reactions both are occurring, hence it is called redox reactions. Electrons are not used by as they passes through the electron transport chain, but are passed on to the pigments of photosystem 1. Hence it is called non-cyclic photophosphorylation. In the diagram you can see the P680 reaction center, okay, made up of more than different pigments. You can see the splitting of water molecule. What does it call? Splitting of water molecule. Splitting of water molecule into, okay, electrons. At the same time, protons are also will be formed while the water molecule is splitting, okay, and half molecule of O2 will be formed. This splitting of water molecule in the presence of light is called photolysis. What is that? Photolysis. Now, the photolysis was explained by Robert Brown for the first time. Okay, Robert Hill for the first time. The photolysis of water was explained by Robert Hill for the first time. Hence, these reactions are called as Hill's reactions. Now, the released electrons from the light harvesting complex will be moving in the upward direction and received by primary electron acceptor in the non-cyclic electron flow called pheophytin. From the pheophytin, the electrons will be enters into the plastoquinone. From the plastoquinone, electrons will be reached to the cytochrome complex. Through the cytochrome complex, electrons will be reached to the plastocyanin. Through the plastocyanin, the two electrons will reach to the photosystem 1 that contain P700 nanometers of light is required for that light reactions. Now, from P700, the two electrons again moving in the upward direction received by the primary electron acceptor pyridoxine. Okay, when the electrons are moving from the pyridoxine, here NADP will be converted into NADPH by an enzyme called NADP reductase. What is that? NADP reductase. Finally, this is a non-cyclic form or a Z scheme. It is also known as Z scheme reaction or non-cyclic photophosphorylation or non-cyclic electron flow. Understood, everyone? Finally, this is the movement of electrons in the Z scheme of light reactions, and you can see the photolysis of water here in the light harvesting complex and formation of NADPH. Okay. Now, after this, simultaneous electrons in the reaction center from PS1 are also excited. So, the whole scheme of electron transfer starting from PS2 to the uphill electron acceptors to the downhill electron transport chain in the PS1, okay, and excitation of electrons transferred to another acceptor, finally downhill to NADP plus for the formation of a reduction of NADP plus into NADPH is called Z scheme. What is that called? Z scheme. Splitting of water molecules. Electrons that were moved from photosystem 2 must be replaced. Here, this is ensured by, basically, this is ensured by electrons available due to splitting of water. The splitting of water is associated with PS2. water is split into two H plus ions, oxygen and electrons. Okay, this creates, okay, so let me show you that equation H2O which undergoes for splitting. Imagine in the presence of sunlight energy it is going to be split, which produces two H plus ions, oxygen plus two electrons will be produced. This creates oxygen, one of the net product of photosynthesis. The electrons needed to replace those from photosystem 1 are provided by photosystem 2. Finally, we need to emphasize here that water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of thylakoid. Then where are the protons and O2 formed like to be released? Okay, so how it will be happens? You can see about the protons and their movement. Already we have discussed in the previous slide. Okay, the movement of electrons in the specific direction. So remaining part of this chapter will continue in the next video. Until stay tuned, don't forget to subscribe.